So the topic I chose to cover for this video was the Krebs cycle and fermentation. So first I'm going to start out with the Krebs cycle. You may have also heard the citric acid cycle, which is abbreviated the TCA cycle. They're both the same thing. It typically takes place in the mitochondria, which is in eukaryotic cells, but in prokaryotes and bacteria who do not have mitochondria, then the Krebs cycle will take place in the cytosol. And what is the Krebs cycle? So it's basically just a sequence of multiple reactions, which is a way to generate energy for the cell by means of aerobic respiration. Basic overview. It's carried out by 8 different enzymes that work to completely oxidize acetyl-CoA into 2 molecules of carbon dioxide in water. And then it also produces 3 NADH and 1 FADH2, and those molecules can pass their electrons into the electron transport chain, which is how the cell can produce ATP to use as energy. And that's really the most important part of the Krebs cycle, is that it produces those 2 molecules who can donate their electrons to produce energy. The pathway of the Krebs cycle. First, before the Krebs cycle can even begin, pyruvate that's synthesized during glycolysis must be converted into acetyl-CoA. So all these metabolism pathways really connect together. So you start with glycolysis, where you're breaking down glucose and you get pyruvate. And then the Krebs cycle is taking the pyruvate from the breakdown of glucose in glycolysis, making it into acetyl-CoA, and then that's going to form your NADH and FADH2, which then converts into the ATP. And then once you have your acetyl-CoA, you can begin your citric acid cycle. The acetyl-CoA will react with a molecule called oxaloacetate, and then that leads into a series of 8 different reactions, which form 6 final products. And then here's your overall reaction. So, which first you get your pyruvate, you make acetyl-CoA, and that also forms an NADH, which goes through the electron transport chain. But you start with acetyl-CoA, NAD+, FAD, ADP, PI, and H2O, which really the most important part of your acetyl-CoA, NAD+, and FAD. But those reactants are going to form 2 carbon dioxide molecules, 3 NADH molecules, 1 FADH2. It forms 1 ATP, or you've also seen it possibly as GTP, which is an ATP equivalent. And then you're going to get 2 hydrogen ions and also the CoA, where it comes off of acetyl-CoA. So, what is the importance of all of this? Like I kind of said before, your major concern with the Krebs cycle are your products of NADH and FADH2, because these 2 products go on to donate their electrons to complexes in the electron transport chain, which is used to generate ATP. And the electron transport chain is a whole other video in itself. But essentially, from each NADH you get about 3 ATP. I think the correct number is actually 2.5, but might as well say 3. 3 ATP formed from each NADH molecule that goes through the electron transport chain after the Krebs cycle, and then you get 1.5, or basically 2 ATP that can be formed from each FADH2 molecule. So if you're thinking 3 NADH total at 3 ATP each, Well, that's already 9 ATP right there. And then you have 2 from each FADH. We said there was 1 FADH produced in the Krebs cycle. So that's another 2 ATP. So you're up to 11 just from your main products from the Krebs cycle. So as you can see, it's really a great way for cells to be able to generate energy. The next thing I kind of wanted to touch on was fermentation, which is the anaerobic way for cells to produce energy. And there are several different types of fermentation, but the most well-known is alcoholic fermentation. And so here's the basic process for alcoholic fermentation. You're going to start with pyruvate, which came from glycolysis, and it's going to be decarboxylated by pyruvate decarboxylase, which essentially is just removing a CO2 group and forming an acetyl group. And then your next step is going to be with the enzyme alcohol dehydrogenase. The dehydrogenase enzyme acts on the acetyl group and it forms CH3, which is then made into your ethanol, which is what everybody is familiar with. And that is your basic overview of the Krebs cycle and fermentation. And then I also want to give Dr. Blair permission to use my video to help future students. Hope you enjoyed.